Good?
Yeah,
Okay, perfect. I will
think
see
so.
you. I'll come in at the end of lecture for the potluck. So see
Okay,
you in a bit.
yeah, see you later. Okay. This is, let me see, can this extend? All right. Cool. And you're on camera, everybody. Okay. So I'm going to lecture for probably 40 minutes or so. So I only will take 15 or 20 minutes during lecture time. And then we can be done and have our potluck. Okay, how are you doing, my children? Last day of the semester. I can't believe it. When do you finish finals? So who was graduating? Kevin, Medora, Isaac, were you all the face?
I'm lucky.
The story.
So back when Tanya was in charge of everything, I got permission from her to have my animal science classes cover some of my food science. And one of them was my capstone.
Oh, where's that? And then capstone was only two of you?
Yeah, it's just me and her.
Wow. Well, I haven't looked at the presentation slide, but okay. Good, so. Chemical or contaminants, residues, and chemical constituents of concern. This is big deal. And every now and then a new chemical shows up and it's of a concern. So we keep updating this chapter. I started this chapter in the 4th edition, got updated in the 5th edition. In the 6th edition, I told the current, and I don't want to do this anymore. Every time I write this chapter, more hazards show up. And I don't want to do it. So we put it on somebody else. Anyway. Okay, so when we talk about contaminants, I want to show this diagram. Oh, I got this here now. Okay, so at any point during the entire value chain, there can be contamination. Let's start with at the land, whether it's animals or plants. What contamination or hazards you might have in your food? Salmonella, like microbial. Okay, let's forget microbial. Same chemical.
Things like fertilizer.
Fertilizers are usually can be organic or natural, but other than fertilizers, hi Lisa, you made it. You're not fired, although you don't work for me. So other than fertilizer, what do you have? So pesticides are added, so there should be a limited amount. There's always a impact action level or a minimum amount that is safe. Pesticide, what do we potentially give our animals when they are sick? Antibiotics. So we give them antibiotics when they have infection. Sometimes they give them low levels of antibiotics so that they get fat with less food. So we don't want antibiotics in our milk. We don't want antibiotics in our meat. Why don't we want antibiotics in our food?
Then you believe that, like, if you eat or like drink enough, you get maybe antibiotics, that you might start becoming resistant or creating resistant bacteria to those antibiotics.
Exactly. So resistant bacteria, especially if you want to process the milk and using fermentation or anything, you might have resistance to certain bacteria. But something else, we might get allergic reaction to antibiotics. So we don't want those in our food. What else can happen somewhere along the way in terms of things we don't want in our food? Pico? Oh, from animal, okay, so that's another chapter, which is extraneous matter. Kevin, you made it? You were this close to being fired. Okay, yeah, that will be another chapter, extraneous matter, like, gonna be very interesting talking about hair and poo, huh?
Like sanitizer, like
Cleaning solutions, yes, chemical cleaning solution. What else? Let me give you hints. Is anybody sensitive to sulfite? For example? No, but we have to put label, if you have sulfite in your food above 10 ppm, we have to put it on the label. So you might also have adulteration. Have you heard of melamine adulteration? I'm gonna talk more about it. What else? What else? You can have during processing, you can produce acrylamide, furans. You can have packaging the bisphenol A. Don't frantically write. It's going to be in the slide. Bisphenol A, for example, is present in plastic bottles. So in water bottles, and this is the carcinogen. So there are so many contaminants that can contaminate. What else we didn't talk about? Fungus, what do they produce? Toxins, mycotoxins. This is a big contamination. So this is a very old one. I should update it sometime. But if we look at this pie, this represents different hazards that were reported in a 4-year period. This part of the pie was chemical contamination. This big part here was mycotoxin alone. We have, of course, the microbiological, we're not going to talk about it. Salmonella is one, for example, but we're not going to talk about it. But also all of the others category that we talked about, fraud, like melamine, biological hazards, extraneous matter will be a different chapter. Ink, cereals sometimes are, before a few years ago, you have a box and you have the cereal in the box directly. Now, what did they do? Plastic bag. So they have a plastic bag because ink was migrated into the cereal, and that is also a hazardous material. Okay. So there are major ones that I'm going to talk about: pesticides, mycotoxins, antibiotics, allergens. What's the biggest recall? Allergens. Allergens are among the biggest recall in the food products. And then we have the chemical additives. We have a whole table summarizing a lot of chemical additives, including adulteration, food additives like sulfide. They act as antioxidants and anti-browning. Nitrite for curing meat. Nitrite with protein form nitrosamines. These are carcinogens. So, and the list is really huge. So let's talk about each big category first. Well, this is a good summary table for you to study from. It's table 31 in your book. It kind of summarizes the main categories that are of concern. So pesticides, mycotoxins, antibiotics. Oh, GMO is another one. Allergens, sulfite, nitrites. And it gives you a quantitative and quantitative qualitative methods. So you can see in almost all of them, you have chromatography. Okay, so HPLC, GC. We have a lot of immunoassays, ELISAs. We have chemical assays, enzymatic assays. But then also we have qualitative similar chromatography, enzyme inhibition, lateral flow strip. That's an immunoassay. So it's good to look at that. I'm going to talk about multi-residue and single residue with the pesticides in a minute. So general consideration, when we want to analyze a sample, these are, not homogeneously distributed. These are could randomly be present in your sample. You remember that from chapter 5, when you have something that could be in different places, you want to make sure that you homogenize your sample. You want to make sure to take a sample that represents the whole population. And you want to take a lot of replication. So remember that from chapter 5. So here, because of the fact that they are not homogeneously distributed, we need to make certain consideration when we take samples, multiple samples. Then we collect the samples, we store the samples, and basically the method we choose depending on the food matrix is high in fat, high in carbohydrate, do we need acid hydrolysis, do we need solvent extractions? What do we know about the analyte? Is it polar? Is it non-polar? Is it ionized? Is it small molecule? Is it large molecule? So we need to know the analyte so that we know how to extract it. Is the method feasible, accessible to us? Are we looking at multiple pesticides, for example, or a particular pesticide of interest? So the method differs, the extraction differs. When you want to look at one, you do more purification. So the steps are lengthy to look at a single residue. So these are the summary of the steps. So sampling, extraction, fractionation, and cleanup. So a lot of cleanup has to happen at concentration so you can enhance the sensitivity of the assay. If you're doing GT, sometimes you want to do derivatization. Then chromatography in many cases, you identify, you quantify, and then you report. So these are the general steps that you want to remember. So here we expand a little bit, preparing the sample, homogenizing, solvent extraction, filtration, and then start your isolation. Once you get, it can be a series of chromatography. It can be solid phase extraction. This very long acronym, you don't have to remember it, but I can tell you what it is. Quick, easy, cheap, efficient, rigor, and safe. I don't know who came up with it. And I don't know how I remember this, but usually the extraction has to be quick, easy, cheap, efficient, effective, that is rigor, and safe. And you talked in gas chromatography lecture, you probably talked about solid phase extraction and solid phase micro extraction. So you want to remember that. And we talked about accelerated solvent extraction when we increase pressure and temperature and enhance extraction using organic solvents. Then we use chromatography. If it's GC, we might use derivatization. Oftentimes, mass spec is added because we need to identify, be able to identify accurately the different compounds. And a lot of time we use immunomass. So this is a good summary to learn. You don't have to remember the acronym here. Just remember solid phase extraction. So pesticides, there are so many pesticides. Hi, Knock, you made it. You were gonna be fired, but you don't work on this. You are. Who's missing? Jason? He's the only one missing now? Okay. This is recorded, Jason. You'll hear this later on. Okay, so there are a lot of pesticides and they keep making more, but originally most of the pesticides were non-polar and can be thermally stable, so we use GC analysis a lot. A lot of the pesticide analysis is based on this. Newer pesticides are polar, thermally labile, so therefore HPLC comes into effect. So depending on the type of pesticide, you can either use GC or HPLC. It has to be registered. In order to be registered, we need to know the limit of use in terms of health? What's the lowest limit or the highest amount that can be considered safe? So in order to determine that and register the pesticide, oftentimes we do single residue analysis because it's very specific to one pesticide. So it has to be analyzed, extracted and analyzed, and determining the maximum level that is allowable in food. So oftentimes, for that purpose, to register pesticides, we do single residue methods, very specific to one residue. Multi-residue analysis, when we have potentially multiple pesticides belonging to the same family, we can extract them, we can analyze them in one assay. So, and then there are semi-quantitative. So what I want you to know is single residue are mostly done to register pesticides. And then multi-residue is usually when you have a class of pesticides that are related, you can analyze them in a single run. So, like I said, mostly it used to be GC because the pesticides were non-polar, thermally stable, and we choose the column based on the characteristics of the analyte. You have a range of detectors that you learned about in with Gary. And then we have internal standards that can be used. Okay, what do you want to remember from this slide? GC for non-polar. You can use multiple colors and multiple detectors. Maybe you want to remember one easy, MS. MS can be used for anything. HPLC is for the newer pesticides that are mostly non-volatile, polar, not thermally stable. Also, we depend on the type of analyte to choose the column. Are we going to use ion chromatography? Are we going to use reverse phase? Are we going to use normal phase? So it depends on the analyte. And the detector, we mostly UV detection, very few are fluorescence, MS. You can remember MS if you want, or UV, what we did in the lab. Oftentimes, we do MS. Why? It cleans up your spectrum. With one MS, you get a lot of ions, a lot of fragments, especially if you're doing GC with electron impact, for example. You get a lot of ion fragments. And if you want to do identification, you want to clean it as much as possible. So you link another MS, we call that MS in tandem, and then you get the ions, the specific ion, and you fragment it a little more, you get less crowded, and then you can have a better comparison to a standard. So this is Etienne, for example, in the standard, and this one in the sample, so you can get a good match. So tandem MS, and sometimes it's MS. 3 MSs in tandem to get to a very good spectrum. ELISA, you remember from last lecture what type of ELISA is this? Is it sandwich ELISA? Is it indirect sandwich ELISA? Is it competitive ELISA? Huh? But
Two antibodies.
what do you think?
So that first one then is.
There's no two antibodies. So it cannot be sandwich. When you don't see your antigens sandwiched between two antibodies, then it's not the same. Competitive, because pesticides are very, very small molecules. They don't have more than one epitope. So it cannot be sandwiched by two antibodies. Large molecules will have multiple epitopes, so you can link two antibodies to them. But very small ones, you have to remember them. Very small ones can have only one epitope. So then you would have the sample, and then you would have the one that has the enzyme linked to it. And then there's competition between the analyte in the sample and the labeled analyte. So the more analytes you have, the less labeled ones will attach to your antibody. I'm reviewing last lecture now. The less will have an absorption. Your absorption will be low. So in competitive ELISA, it's inverse. The higher the concentration in your sample, your F-line, the lower is your absorption because the labeled one will bind less. There will be higher competition from sample. So in sandwich ELISA, direct and indirect, as absorption increase, concentration is higher. Here is the opposite. We can do thin layer chromatography to screen. We can also, in the thin layer chromatography, what they do, you run your samples, the different samples and standard. Usually pesticides inhibit a specific enzyme, like say urease. So what you do is you separate your sample, you incubate it with the enzyme solution, and then you add the substrate. So all the plate will turn color, except where you have an pesticide. You will have zones that don't have color because those pesticides inhibited the enzyme. So that's a qualitative way of determining presence of pesticides. Mycotoxins, fungal. Most common is Aspergillus flavus. These toxins are harmful in terms they are carcinogen, and then they might be genotoxin. If they are a genotoxin, there is no allowable tolerance level, especially aflatoxin B1, I believe, there is 0 tolerance level. Some other aflatoxins, you have tolerance level less than 15 parts per billion. How do they get on the food? Fungal growth. So sometimes you can see the fungal growth, sometimes you don't, but that if you don't see it doesn't mean you don't have the toxin. There are different types of toxins, different characteristics. The most common chromatography is HPLC, reverse phase HPLC. They're polar, relatively polar, they're non-volatile, so reverse phase HPLC is one of the most common ones. You can use UV or fluorescence. You can also use MS. I don't have it here. You can also have competitive ELISA, same principle, small molecule. You can use competitive ELISA, and you can use thin layer chromatography. You can use immunoaffinity chromatography, very specific antibodies for the mycotoxin. So, you know all these methods now, it's just application in this case. Antibiotics. So they are given to animals at therapeutic level. If the animal has an infection, so they get antibiotics. Sometimes they give them at sub-therapeutic levels. That means just a little bit to prevent infection and to make them grow fat with less food to gain weight. However, if the animal is getting ready to be slaughtered in like a few days, they have to stop the sub-therapeutic levels so that there is 0 antibiotics in the food, in the meat or the milk. No tolerance level could be 0. So remember that. Tolerance level is 0 for antibiotics. Why? Because some people have allergies to antibiotics. Another issue is bacteria get resistance. We said that already. So the hypersensitivity reactions, and then if we are doing any fermentation and we need production of flavor or acid, then the presence of antibiotic will impact the processing. So there are quantitative and qualitative way of measuring. The most common one is a very qualitative, used for milk. It's just, you take, very easy to do. You take a plate, specific media for the particular bacteria, Bacillus stereothermophilus. you take a little disc here, you don't probably see it, there's a little disc that you have your milk sample in, and then you incubate. Then you will see that there are growth where there is no disc, and there's zones where there are no bacterial growth. That means there is antibiotic in these samples. If there's no antibiotic, you'll see bacteria growing everywhere. Charm test is a very common test that you can find commercially. It's very easy. It uses radio-labeled antibiotic. And these antibiotics will compete for bacteria that you add to your sample. You add a bacteria, and then the radio-labeled drug will compete with any drug present in your sample. The more antibiotics present in your sample, the less radio-labeled drug will bind to your bacteria, so you'll have less bleeding. So again, there is competition here. The lower the reading of the radio-labeled drug, that means you have more of the actual drug in your sample. So, quantitative methods, reverse phase HPLC antibiotics, very common with HPLC. You can also, for some antibiotics, you can do GCMS. But with both, you use MS as a detection. Please remember, this is a very important note for your final. Sometimes I ask you, what is the method and what is the detection? So the method of analysis, you would tell me HPLC, GC. When I tell you, or GC, you pick one. And then when I ask you what is the detection, then you would go MS, UV detection, refractive index, whatever. That's your detection method. So, method of analysis and detection, two different things. You want to know that. Okay. Allergies. Okay, the list is growing. Now it's big 9. It used to be big 8. Sesame was added. In a couple of years, I bet you that there's going to be P there, because there's a lot of pea protein now in a lot of foods. 50 years ago, there was no soybean as an allergen, but with more exposure, especially in the North American and Europe diet, then this became an allergen. So this list is growing. We should label based on Food Allergen Labeling and Consumer Protection Act. Even if, let's say, you don't have peanuts in your M&M, you have to put on the package, processed in a facility that utilizes peanuts, has to be put on the label. So there's strict labeling requirements to list all the ingredients by their common name. People need to know soybean, you have to put wheat. You cannot use the term that's coming from wheat and soybeans that the consumer cannot recognize. So you have to use common names. Methods of analysis. Most common is protein methods of analysis. ELISA is a big one. Western blot for qualitative, and then biosensor amino acids. So, but these methods have a flaw in them. When we analyze for protein, we have to extract the protein from the food matrix. And depending on the method of extraction, we may or may not extract the allergens. So here's an example from the figure in our chapter. So this is a sample that was extracted using salt extraction, adding salt to the buffer. Here's a sample where we didn't add salt to the buffer. So globulins, if you ever have learned about cereal protein fractionation, globulins are salt soluble based on osmotic fractionation. So if we have globulins in the protein and we don't use salt, they don't show up. So there are potential allergens that we cannot detect. So that's the drawback in that protein method. We have to make sure that we're able to extract all possible proteins that could be allergen. The other methods is determining the presence of DNA of the particular allergen using PCR. I'm not gonna go into that. I'm not familiar very much with PCR. I just wanna remember that there are DNA methods using PCR. But now the drawback of that is when you use a protein isolate, for example, in your formulation, that protein has been isolated from the original matrix, from soy or wheat or what have you, then you lost DNA. There's no longer DNA in that sample. So if you do PCR or we need it, you won't able to get that. So both methods have their pluses and minuses. So we have to determine which one would be appropriate for the type of sample we have. Okay, here's the wonderful list that made me quit writing that chapter because it keeps increasing. And every time there is an increase, that chapter gets bigger. So, but we have important ones that I highlight in the next slide. Sulfide, nitrites, melamine, packaging material, and acrylamide can be produced during processing. Also, monofluoropropane can be produced during processing, and then we have benzene that can be present due to different ingredients that you use, and perchlorate, chlorate contamination from soil that might have fuel material in them. There are others, and different countries have different regulations. Some countries ban certain components. Some countries have legal limitation in terms of concentration. And there are some components known as international contaminants. Some are approved for use, but of concern, like monosodium glutamate, might cause headaches in certain individuals. And then natural constituents, these are produced during processing. So things could be there because they were added or contaminated. The sample got contaminated. Some are produced during processing. Some are added here for to prevent browning in dried fruits, for example, to prevent oxidation, antibiotics, antimicrobial agents. These are processing tools for curing meat, nitrites. So depending on what product we're working with, we can have any of those in them. So now I'm gonna give you a briefing of all of them. So, but here's a general overview. These compounds are present, could be present in a very small amount. They're very unique compounds, so determining their presence is tedious. That means the methods of analysis are long, tedious, expensive. Most of them are chromatography and mass detection. And they have to be very sensitive because we need to determine them at very small concentrations. So limit of detection is a big concern. How low can we go in identifying them and quantifying? The methods keep developing. More methods come up to make sure that we can analyze them quicker, have lower limits of detection, are simpler to use, and so on. So very quickly, I'll go over each one of those and I'll tell you what you need to know. You need to know why sulfides can be present in foods. So they can be present naturally in wine, or you can add them to prevent microbial growth and browning. So you want to know that. Why sulfides can be present in food. So you want to know or have an example. Why we don't want it? Because some people have sensitivity to it. we have a requirement. You don't have to remember that number, but there's a labeling requirement. If the product has greater than 10 parts per million, you have to put containing sulfide. There are a lot of methods to detect sulfide, but sulfide can be present in so many different forms. Bound, not bound, free. So there is not one method that can cover all forms, but there is one method that is commonly used. You want to remember Monnier-Williams method. That's the method that the FDA referenced. It measures total sulfur dioxide that is free and some bound. So in order to determine the sulfides, and in the form of SO2, you heat with HCl. So HCl is going to convert all of your sulfide or most of your sulfide into sulfur dioxide. From there, you oxidize it with peroxide to form H2SO. So what do I remember from here? Acid to get SO2, SO2 oxidized, peroxide, you don't have to remember it, but say oxidized, to give you sulfuric acid. And then sulfuric acid is either measured by measuring turbidity or gravimetrically by weight. So these are the key things you want to remember. Now, there are other methods for sulfide, but I would say FYI for this. Don't have to remember. If you remember Monnier-Williams, that's good. But if you want to know, there are so many different methods. An enzymatic method, for example, HPLC as well, and titration. So there are different methods, but the FDA one refers to Monnier-Williams. Nitrites, I said they're added as processing tools to cure heat. So you have nitrate, NO3, and you have nitrite, NO2. So it acts as antimicrobial. It gives you that pink color of your cured meat. However, there is legal limitation. If you have nitrates, and you have protein in your sample, you're going to form nitrosamines. You're going to form nitrosamines right there. So these are carcinogens. So we don't want them. We don't want to consume a lot of them, basically. Avoid too meat. Method. It is colorimetric method. Don't have to remember any other than just say colorimetric method. And or chromatographic method. Iron chromatography with conductivity. Don't have to remember in the middle here, but it could be colorimetric method or assay or HPLC using ion chromatography. Rapid method, the ion selective electrodes that Dan talked about in the minerals section. Test strips as well. So rapid methods. Don't have to remember the concept here. Melamine, I'm almost done. Melamine is the contaminant that was added to enhance protein in the food. They did it as adulteration, but when they did it, they didn't know that it can be fatal. So it caused kidney problems, and that caused potentially death. So they found it in milk products. They also found it in imported pet food. So there were cases of pets who died eating specific pet food. So now, why did this pass as a protein?
Because the analytical methods that are used are based off of nitrogen.
Yes, great. So Carol, you're on the money. So you have amine groups, you have a lot of nitrogen. So when we do Kelda or LEAP or DUMAS, the main official methods of analysis, these are gonna be converted by the conversion factor to protein. So you don't have more protein in the sample, you have more nitrogen due to this adulteration that reads as protein. So they were tricking people that you have higher protein in the diet of pets or babies, where it's not protein, obviously. So chromatography is the method with mass spectrometry, is the method for identification and detection. There are other methods that you can use for qualitative, for example, like NIR and mid-IR. And there are immunoassays. But if you want to quantitate, the best way is chromatography coupled with mass spectrometry. Bisphenol A, it is primarily used to produce plastic. It can leach from the plastic bottle into the beverage, have health effects, especially for babies and young children. It's banned in some countries, and some countries allow certain limits. Again, chromatography, you can see DCLC mass spec in tandem with high sensitivity and very low limit of detection. And of course, immunoaffinity columns and ELISA as well. For methylbenzophenol, this comes from ink. So, and that's the example of cereal box, and now they are in plastic container to avoid any leakage of this chemical from the ink on the cover, on the outer package to go into the food. So long-term health risks for children, impacting specific organs, and then method of analysis, again, LC and F. You don't have to remember limit of detection. Furans that are produced when we are processing food that have high carbohydrate, potentially some acid and heat. So these can be produced, and these furans are carcinogens. They are volatile, so they can be used. You can use headspace PCMS, where you put the sample. The furan is volatile, so it will be on the headspace over the sample. You take a sample of that headspace compartment above the sample, and you do PCMS. Acrylamide also can be produced in food. For example, deep fried potato. You have, or chips, okay? You have carbohydrates, and then you have a protein, especially an amino acid aspartate reacts in a Maillard reaction to give you acrylamide. So aspargine reacting with a reducing sugar at really high temperature gives you acrylamide, which is carcinogen. Again, LCGC coupled with MS. Benzene can be also, it's also carcinogen, but if we have a beverage that contains ascorbic acid, that's vitamin C, with benzoate salt, they can be formed. It's volatile, so we can do headspace GCMS. Monochloro-propane. This is specifically formed in a protein ingredient that is hydrolyzed by acid. So soy protein will have a very small amount of fat, residual fat. In the presence of acid hydrolysis, we can form monochloro-propane, especially if we use HCl as the acid for hydrolysis. It is suspected, it is carcinogen and suspected to be genotoxin, so we don't want it at all in our food. GCMS is the method. Finally here, I think, chlorate, this is, component of rocket fuel. It can be in the soil, so it might make its way to water, to milk because of, cow feeding on certain, grazing and then getting that from the soil. And it was reported in Lect 21. Iron chromatography, so basically HPLC using iron chromatography and mass spec. So this is the table that would be helpful to study from. It is crowded, you have it in your book, you can see it in bigger. But really what I might ask you is, what is that compound and why did it make it into the food? So we went over all of them and how it came into the food. Why is it of concern? Most of them are carcinogens. You can see here. One thing I didn't mention is heterocyclic amines. This has been recently, in the past 10 years or so, determined to be harmful. And don't overcook your meat. If you're having a cookout, and don't eat burnt meat, because it will have high amount of heterocyclic amines that are carcinogens. So based on the protein, specifically creatine and maillard reaction. So what food they can be in, why they are there, why they are of concern, mostly carcinogen, you can see. And most of the methods are LC, GC, and MS. Okay, so I'll end here. There's just a small part on extraneous matter I'll cover in class before we have our potluck. I'm gonna stop the recording now. No, you can't be tired yet.
Take back, please.
That's okay.
It's these chairs.
What's that?
It's these chairs.
The chairs that you're sleeping?
Yeah, there's something about it, it's just like, feeling my back.
Oh, I'm so sorry. Yeah, we have one chair with a back to it. You can fight over it if you want.